Hi everyone, welcome to Micro Mahasutra, and we are in 6th session of our discussion, and in this session we are going to discuss about the general mycology. Mycology is nothing else but the study of fungus. Okay, so the beautiful mushroom which you eat is nothing else but a fungus only. So in general mycology, mostly I am going to discuss the relevant clinically important points, right, which are used for formation, for manufacturing of antifungals. Basically, we will touch those parts only. So this is going to be an integrated session with the pharmacology and medicine, of course. So please remember fungus. See, the fungus is a eukaryotic organism, whereas bacteria, viruses, they are prokaryotes. But just like bacteria, fungus is also a heterotroph. Okay. Now please understand, what are the different mechanism of actions of antifungals? So, in order to understand the mechanisms of antifungals, let's quickly see this. So, see here, first of all we have to understand that the fungus has got a cell membrane, and in the cell membrane there is E. What is this E? E is ergosterol. What is E? E is ergosterol. So, ergosterol is a vital component of cell membrane, okay? And remember like that, ME for ergosterol. And in order to synthesize that ergosterol, we require two enzymes. Fungus requires two enzymes. One is this one and second is this one. What is SQE? So, remember, squalene epoxidase, that is called as SQE. So, squalene epoxidase is the enzyme which is required to synthesize ergosterol, and also 14-alpha-D-methylase is the enzyme which is required to synthesize the ergosterol. But if we want to destroy the cell membrane, what we'll do? We will inhibit both of these, squalene epoxide and 14-alpha-D-methylase. How? In order to inhibit squalene epoxidase, the antifungal which is used is called as allyl amines. It is called as what? It is called as allyl amines, that is going to destroy squalene epoxidase. And then you have got azoles like ketoconazole. They are going to inhibit 14-alpha-D-methylase. So both allyl amines and ketoconazoles are going to inhibit squalene epoxidase and 14-alpha-D-methylase. That means no ergosterol will be synthesized. Because ergosterol is not being synthesized, that means the cell membrane is not being synthesized. Just tried a breathless version by 𝗖𝗵𝗵𝗿𝗺. Okay. So cell membranes are not going to get synthesized. Okay. Similarly, there is an antifungal called as polyene. So what polyene do? Please understand that ergosterol, in order to, once ergosterol is synthesized, this ergosterol has to bind with the cell membrane. If you will give polyenes, that's an antifungal, if you will give polyenes, these ergosterol will not be able to bind properly, and because it is not binding properly, it is going to increase the permeability of the membrane. So polyenes bind to already synthesized ergosterol and disrupt its interaction with the cell membrane, increasing the membrane permeability. So anything, polyenes are going to increase the membrane permeability. So that is why with any drug, any antifungal drug, we combine polyenes so that that drug can penetrate better, because it is increasing the membrane permeability, no? So the other drug will go better. For example, you combine polyenes with ketoconazole. So polyenes increasing the permeability and ketoconazole can go better. So it will show what? It will show better penetration. Okay. Coming on to next part, 1,3-beta-glucan synthase. 1,3-beta-glucan synthase. Now please understand this. 1,3-beta-glucan synthase catalyzes the synthesis of glucan. Catalyzes the synthesis of glucan. What is this glucan? So remember, fungus has a cell wall, and the cell wall is made up of Mannans, glucan, chitin. It is made up of Mannans, glucan, chitin. And this Glucan synthesis requires 1,3-beta-glucan synthase. So there is a group of antifungal called as echinocandins. They are going to inhibit this enzyme 1,3-beta-glucan synthase. Echinocandins inhibit 1,3-beta synthase, inhibits glucan, inhibits the cell wall synthesis. Also, fungal cells have got cytosine deaminase. They contains a enzyme called as cytosine deaminase, which converts flu cytosine to some products that are going to interfere with DNA and RNA synthesis. So because there will be no DNA RNA synthesis, there will be no fungal cells. Coming on to the morphology of fungus. So either the fungus is a mold or the fungus is a yeast. Either the fungus is a mold or the fungus is a yeast. These are some morphological forms in fungus. So mold means when there is a multicellular fungus, this is mold. If it is a unicellular fungus, it is a yeast. Okay. So fungus can be mold, can be yeast, but there can be some dimorphic fungus also. Dimorphic fungus means mold and yeast both, depending upon the temperature, either mold or yeast depending upon the temperature. What does that mean? Remember, if it is cold, the dimorphic fungus will be mold. If it is heat, dimorphic fungus will be yeast. Okay. So simply speaking, a multicellular fungus is a mold, a unicellular fungus is a yeast, but dimorphic fungus can be mold or yeast. Mold if it is cold, yeast if it is heat, depending upon the temperature. Coming on to what is hyphae. Remember, this is the multicellular organisms, multiple cells. Okay, so this is a filament which is made of those multiple cells. So hyphae are long tubular structures formed by multiple fungal cells lined up end to end. Can you see? Yeah. And what is yeast? So normally yeast is like this, and this is the budding. These are the buds which are shown, and after some time this bud will cut off and will grow as a new fungus. But for example, this is going to grow, one bud, other bud, but they are not getting cut off. So there will be a similar structure like hyphae produced, but that's not true hyphae because the fungus is unicellular. So that's why it is called as pseudohyphae. So what is pseudohyphae? Pseudohyphae are formed when bud fails to break off the original yeast cell, forming the long chains which resembles the hyphae. Okay. What are the dimorphic fungus? Remember, in order to remember the names of dimorphic fungus, you have to remember STS as the mnemonic. And if you don't know STS means State Transport Services. So S for Sporothrix, T for tularemia, S for systemic mycosis. Sporothrix, can you see? This is Sporothrix. Then T for tularemia, which they have not written, but yeah, it is also dimorphic. And then you have got systemic mycosis. What are the examples? One is blastomyces, next is histoplasma, next is coccidioids, next is paracoccidioids. Okay. Also, Candida is a dimorphic fungus. Okay. So remember, C and G always go together, they are similar. So G for germ tube, right? So germ tube is formed, Candida is the one which forms the germ tube. Remember, germ tube, germs, oh my god, it's a high alert. So germ tube is formed at high temperatures. Okay, germs are high alert, no? So they are formed at higher temperatures. Okay, so Candida exists at pseudohyphae and budding yeast and pseudohyphae. So it is also dimorphic, na? Two morphologies. So yeast at 20 degree, but germ tube formation at 37 degrees. Okay. Now hyphae after some time grows as a single individual fungal cells within the hyphae by the help of mitosis. So what I am trying to tell you, that this was the initial parent which gave two cells with the help of what? With the help of mitosis. Okay, they are going to divide. So it leads to the formation of mitotic spindles. So if somehow we stop the mitotic spindles, the fungus will not be able to grow. That's why there is a drug which is called as greaseofulvin, G for greaso, G for growth, growth inhibitor. Greaso means growth, fulvin means failure. So it leads to growth failure. Why? Because you are going to inhibit the mitotic spindles. So it leads to inhibition of mitosis. That means there will be no growth. Okay. So please keep the greaseofulvin like people away from you. Then only you can grow better in your life. My comment section is always there for you, for your blessings, love, care and support, and definitely the feedbacks. Thank you so much for this session.